Hello everyone, here onwards I am going to continue next part of carcinogenesis. From my previous video you have learned what is the function of p53 gene and how it diverts a cell in repairing process once it is detect any abnormality of that particular cell, and if it is cannot successfully repair, how it is diverts into apoptosis or permanent cell cycle arrest. Here also you can summarize that. Here is a normal p53 gene. When there is DNA damage, hypoxia or cell cycle abnormalities detected in a cell, p53 gene is activated and further proliferation or further cell cycle is arrest at that point and divert that cell into repairing process, and after successful repair the cell cycle restarts and that cell can undergo the cell cycle. So if that cell is not able to repair that abnormality successfully, p53 diverts that cell into permanent cell cycle arrest or apoptosis, means programmed cell death. So imagine now p53 gene is mutated. So if there is any DNA damage, hypoxia or cell cycle abnormalities is there in a particular cell that is undergoing cell cycle, because the p53 gene is mutated now, this p53 gene cannot detect that particular abnormality in the cell. So this mutated p53 gene does not arrest the cell cycle then and there. So what happens is damaged cell continues to divide in the cell cycle. It continues the cell cycle, which may result in a cancer. So if there is a normal cell and after DNA damage occur, if it is failed to repair, there is mutation in somatic genes, and there are four basic steps of carcinogenesis. So as you can see here, when there is a mutation in the genome of somatic cells, there are four basic steps that occur in carcinogenesis. First one is activation of growth promoting oncogenes. Examples for growth promoting oncogenes are, as you can see here, RAS, MYS and HER2 gene. Action of these oncogenes are, they have unregulated cell growth. When there are these oncogenes present, there is unregulated cell growth. Actually they promote cell growth. Normally in our body there are proto-oncogenes, and they are well regulated by growth factors. That means they have initiators, well regulated initiators and well regulated feedback mechanisms. So if those proto-oncogenes are mutated, cells have overproliferation. And also oncogenes are considered as dominant genes. They are considered as dominant genes because if there is a mutation even in a single allele, can cause proto-oncogenic effect. Those oncogenes, the examples are RAS, MYC and HER2 genes, encode for transcription factors and growth regulating proteins, and they encode for proteins involved in cell survival, as well as they encode for proteins involved in cell-cell or cell-to-matrix interactions. These cell-cell or cell-to-cell interactions are important in tumor invasion. You already know in tumor invasion, it is first invade the cellular matrix and then it invades the basement membrane of blood vessels and go into circulation. Therefore, these oncogenes encode proteins involved in cell-cell or cell-to-cell matrix interactions, which is important in tumor invasion. There are some examples for oncogenes. There is excess production of HER2 receptors or HER2 genes in breast cancers, gastric malignancies. Mutated RAS receptors you can see in lung malignancies and colonic carcinomas. MYC and BCL2 can be seen in lymphomas. Also, ERBB1 receptors are overexpressed in squamous cell carcinomas in lung and glioblastomas. So those are the examples for oncogenes. You have to remember those: HER2, RAS, MYC, BCL2 and ERBB1 receptors. This HER2 receptor also called as ERBB2 receptors. Then the second step of carcinogenesis is inactivation of tumor suppressor genes. Inactivation of tumor suppressor genes. That means TP53 and RB genes. Those are inactivated. I told you earlier as well, P53 gene can detect any abnormality in cell cycle and it can divert that particular cell into repairing process or permanent cell cycle arrest or then apoptosis. So these tumor suppressor genes are very important to prevent uncontrolled growth of cells or tumor genesis. They prevent uncontrolled growth or tumor genesis. So imagine if these tumor suppressor genes are mutated, then there can be uncontrolled growth. We cannot control the growth of that particular cells. The other thing you have to remember is the tumor suppressor genes are divided into two parts as two types as gatekeeper genes and caretaker genes. Caretaker genes are the genes involved in DNA repairing process, maintaining genetic stability and minimizing the rate of mutations that occurs in cells. Gatekeeper genes again can be divided into two types as governors and guardians. The gatekeeper genes do regulate cell growth and prevent cellular proliferation. So there are two types of gatekeeper genes, governors and guardians. Example for governors are RB gene. Examples for guardians are TP53 gene, which encodes for P53 gene. This governors or RB gene important to inhibit cellular proliferation further. That means they prevent excessive cellular proliferation. So, if there is a mutation in this RB gene, there can be uncontrolled cellular proliferation. The function of these guardian genes or TP53 genes is sensing of genomic damage. Its normal mechanism is sensing of genomic damage. But if this gene is mutated, genetic materials can be passed to next generation as well. And also you have to remember that in tumor suppressor genes, both normal alleles of tumor suppressor genes must be damaged for tumor genesis. That means to become a tumor, both alleles of tumor suppressor gene should have been damaged or mutated. Then the third step of carcinogenesis is alteration of genes that regulate apoptosis. Alteration of genes that regulate apoptosis. The gene that responsible for apoptosis is BCL2 gene. So mutations in these genes that regulate apoptosis cause absence of programmed cell death. That means those cells are resistant to cellular death. Therefore, even though there is any abnormalities detected in that particular cell, because that cell cannot be undergo cellular programmed cell death, that particular abnormality or that particular abnormal cell can undergo cell cycle, enhancing cellular survival. So, next step of carcinogenesis is defects of genes involving DNA repair. So here you can see if there is activation of growth promoting oncogenes and inactivation of tumor suppressor genes, it leads to unregulated cellular growth. And if there is alteration of genes that regulate apoptosis, they decrease apoptosis or there is absence of programmed cell death. Then those factors lead to clonal expansion and tumor genesis.